Hello and welcome to GeneLab Chats. Today we will be interviewing Thomas Cahill from Queen's University Belfast. The space environment poses many challenges that can induce significant physiological changes to all species. One of those physiological changes is muscle atrophy, which can negatively impact the health of astronauts. In his recent publication, Thomas reanalyzed 7 transcriptional profiling datasets from the GeneLab database to detail the mechanistic insights on mammalian muscle responses to spaceflight. In this interview, he will discuss this research and highlight how GeneLab's analysis working groups helped highlight his work. Welcome, Thomas.
Thanks for having me.
Let's first start off by asking you to please let the audience know what were the most important findings in this study.
So in this study, we first set out to characterize the response to spaceflight in slow and fast twitch muscles. And in the fast twitch muscles, which are more atrophy resistant, we've seen a larger immune response, increased proliferative gene sets, and decreased PDE genes, which regulate cyclic AMP, which is the signaling molecule involved in muscle growth and regeneration. Whereas in the slow twitch soleus muscle, which is more prone to atrophy, we've seen a smaller immune response, the negative regulation of proliferative gene sets, and an increase in the expression of PDE genes, suggesting decreased cyclic AMP signaling. We next compared ground-based models with space-flown mice. So in a hind-limb unloaded model of the gastrocnemius, which is a mixed fiber type muscle, but more functionally similar to the slow twitch soleus, we observed a phenotype more similar to that of the fast twitch EDA with an upregulation of proliferative genes. While the space-flown muscle response was more similar to the functionally similar slow twitch soleus, displaying increased tumor suppressor genes. And then sort of in line with previous research showing that muscle responses group by functionality in spaceflight. We also compared a human bed rest quadricep muscle with space-flown quadricep muscle in mice, which showed shared results relating to glucose uptake, the extracellular matrix, and muscle dystrophy. But I think one of the main takeaways from the paper is demonstrating the advantage of using smaller model organisms and modeling the mammalian response to spaceflight. When we compared the response to spaceflight in C. elegans with the slow and fast twitch mammalian muscles, we've seen a phenotype more consistent with that of the fast twitch muscle. So, for example, we've seen a shared upregulation of proliferative gene sets, as well as genes involved in the unfolded protein response, which were downregulated in the soleus, as well as seeing the downregulation of PDE genes, suggesting increased cyclic AMP signaling. However, when comparing the Drosophila with the slow and fast muscles of the space-flown mice, we've seen that the response in larval Drosophila was more like that of the slow twitch muscle, displaying increases in protein metabolism, a decrease in unfolded protein response genes, and a downregulation of proliferative gene sets. So I think that in using these smaller organisms, which are easier to maintain, share considerable genetic similarity with humans, especially in disease-causing genes, and they're often cheaper to send into space, then we can expand the breadth of space-flown data sets, which will help us sort of address knowledge gaps and generate and test new hypotheses.
That's absolutely fascinating. Thank you for that summary. One last question here is, how did the GeneLab database and analysis working groups help assist you in your work so you can achieve your goals?
So NASA's GeneLab database has made space biology datasets publicly available, as you know, which increases their accessibility to the scientific community. And the AWG has also developed analysis pipelines to optimize the processing of raw omics data, which increases the comparability between datasets. So these things allow us to maximize the scientific output from the limited number of spaceflight datasets that are currently available. But being a member of AWG allows you to collaborate internationally with around 150 different scientists and benefit from access to their specific skill sets and expertise when working on projects. So I would encourage anyone that's interested in space biology to get involved because they put out some really interesting research. For example, showing the response to different doses of radiation with different types of radiation across the animal kingdom. Or the multi-omics paper, for example, which included analysis of the NASA twin study, which revealed the role of mitochondrial dysfunction as a central player in the response to spaceflight, and they've always got something really interesting going on.
Well, thank you so much, Thomas. That was incredibly informative.
Thanks again.
And for all of those watching, you can learn more about this work, how to join the analysis working groups, and access all the data sets analyzed in this work with the links provided in the description section below. Thank you.